So as Steve said, my name's Dominique, and this is my exit seminar for the Horticulture and Agronomy program here at Davis. I did both my master's and my PhD with Steve in the breeding program, strawberry breeding program here at Davis. And so I'm going to be talking about the projects I've undertaken during my time here. So these projects encompass the genealogy of the cultivated strawberry and host resistance in Strawberry-Vascular Wilt Pathosystems. Oh, there we go. So the general outline for this talk, I'll start with the genealogy of strawberries, since that is its own separate and rather unique topic. And then I'll start talking about the disease resistance work that I've done, specifically on verticillium wilt and fusarium wilt. Now, because of the breadth and scope of these projects, I'm not going to be bogging you down with every single result. I'll be hitting on the highlights that have come out of these projects. However, I do encourage you to go look at the publications associated with these after the talk if you're at all interested. The genealogy of strawberry and verticillium wilt projects have been accepted and or published, and so they should be ready to read very soon or immediately. And the fusarium wilt project, we are working on getting that published soon. So starting with the strawberry genealogy, I think I need to really start out by telling you about the cultivated strawberry. So this is Fragaria ananassa, which is an octoploid interspecific hybrid. It was hybridized about 250 years ago in Europe between Fragaria virginiana, imported from North America, and Fragaria chiloensis, imported from South America. Here you can see a population structure analysis of the greater Fragaria diversity. Fragaria chiloensis, one of the progenitors, has a yellow cluster here. Fragaria virginiana clusters down here and overlaps with the heirloom cultivated strawberry. But I do want to focus first and foremost on this UC Davis material, where it goes blue to red or past to present. And so this is where the genealogy project really started. When we look at UC Davis, the UC Davis strawberry breeding program, it has been hugely influential to the strawberry community as a whole. It started in the 1930s with Thomas and Goldsmith and has continued ever since until the modern day. But while I have the pictures of these men up here, they are certainly important in terms of the decisions they made to guide the breeding program to the success it is today. It really is the plant material, which is the star. When you look around the world, 60% of the strawberries harvested come from UC Davis varieties, which should attest to the success of that plant material on its own. But you also have to consider that we not only have commercial blockbusters like the Camarosa cultivar, which was one of the most widely grown cultivars in the world, but you also have pioneering material, such as the material that introduced the day neutrality trait to the industry. But when we look at this plant material, we do find that we lack a lot of key information about it. Frankly, over the years, we have lost a lot of information regarding the parentage and the breeding history, whether that is in the loss of the breeding notebooks of Thomas and Goldsmith and Bringhurst and Voth, or the loss of pedigree records due to technical difficulties, which is obviously a much more modern program with computer databases. And so that leads to the main motivation for this genealogy project, which was to document the collection, make sense of the breeding history, so we can then be good stewards of it. Another motivation here was to place in context the UC Davis material against material from around the world. So in order to do this, we constructed a very complete pedigree database. We started off by rescuing records from computer databases. We would like to thank Robert Kerner for his contribution there. We also assigned parents to individuals who had no parental information. For that, we used DNA forensics and SNP exclusion analyses. Mitchell Feldman was the one who spearheaded that part of the project, so I'm going to shout him out there. But we also managed to track down those physical records that had walked away from the university all those years ago, namely the breeding notebooks of Bringhurst and Voth and Thomas and Goldsmith. We also scoured the germplasm databases out there for pedigree information, so not only North American germplasm, but European and other germplasm from other countries. And we pulled down pedigree lists from published literature, academic theses, bulletins from agricultural stations, and narratives that had examined the breeding history of strawberry. And finally, we did go into the patent databases and pull pedigree information down from there. And so in the end, we ended up with more than 800 individual sources for this pedigree database, where each source generally contained more than one individual pedigree. During the process, we also collected passport information, such as the birth year, the geographic origin, breeder name, species, and more. And so what did we end up with? Well, we ended up with this really interesting and I would say complete look at the global strawberry genealogy. This pedigree extends from before 1800 to the modern day and contains information on 8,851 different accessions. You could also further subdivide this into the California genealogy, which leads to the UC Davis program, or the cosmopolitan genealogy, which is everything else. So we were very happy to have this resource and decided we were going to leverage it to do some really cool analyses that haven't been done in strawberry before. And again, I would really want to emphasize that I'm hitting just the highlights here. There are so many more cool results and takeaways that will be in the final paper. One of the really cool analyses we did was a population structure analysis of the California and the cosmopolitan genealogies. Here on the left, you can see the actual pedigree networks, so how those pedigrees look like. And then to the right, you can see a population structure analysis. And the population structure analysis, for your information, was done on a pedigree relationship matrix, which had been augmented with genomic information, so a pedigree genomic relationship matrix. So on top here, you can see the genealogy of the California population, and you can see that there is a strong chronological trend, where dark red is the distant past and dark blue is the present. And you can see indications of bottlenecks that we know existed here in both of the pedigree network and in the population structure analysis. On the other hand, the cosmopolitan genealogy showed heavy admixture between geographies. You can see here in blues the North American countries, green the European countries, and red the Australasian countries. And while these geographies are very far apart geographically, you have seen a lot of migration and mixture between them. Another level of analysis we can do is at the individual level, looking at every single session for its own, on its own weight. For this, you could assign metrics that any plant breeder should know, such as genetic contribution, or the degree to which a single ancestor has contributed to a focal population. But you could also use more unique metrics such as betweenness, which is a metric generally used in social network analysis. This is sometimes also called bottleneckness in other fields. And so this measures the degree to which a single ancestor has controlled the flow of information through the pedigree, or how much it has acted as a bottlenecking accession. You could also use a metric like out degree, which is a direct measure of how prolific an ancestor has been as a parent. Here is an excerpt table from the paper in which we looked at these metrics for the most prominent and historically important ancestors of cultivars, and we were very happy to see that our these metrics and what cultivars they picked out lined up with the narrative history of strawberry that does exist. What was important in those narrative histories was important in these results. And that showed that we could be really confident in these sort of analyses. We could also look at the evolution of these populations over time for certain measures. For example, we could look at the degree of inbreeding over time for both the cosmopolitan, shown here in the dashed, or the California population, shown here with the solid lines. So in both populations, you've seen a steady increase of inbreeding, but more inbreeding in the California population. But when you think about that, that is expected given that that contains the UC Davis breeding program, which is a single breeding program, which is insular and hasn't seen much migration in the near past. You could also look at something like the percent of the genetic contribution which comes from wild species. So in the pedigree records, we did find that there was repeated hybridization and migration with those progenitor species, Fragaria virginiana and Fragaria chiloensis, in the early part of the pedigree. However, as domestication ended and improvement began, you started to see Ananassa being used more, you saw less of these wilds come in, and of course, the percent wild genetic contribution began to decrease. And so that is also not an unexpected result. And I do want to emphasize the importance of the passport data because that is a key part of this whole pedigree database. It can let you do interesting analyses that you wouldn't be able to do otherwise. For example, we looked at the average selection cycle length across the entire genealogy and found that on average, it takes about 16 to 17 years. However, that has decreased consistently over time, suggesting a broad acceleration in genetic gain. And so I do want to highlight that the most important thing about this pedigree database is that it is a really thorough clarification and detailing of the breeding history of the modern day cultivated strawberry. And it does provide a really cool tool to the broader strawberry community. Again, I really do encourage you guys to go check out this paper if you're at all interested. I could not hit on even half of the results and cool takeaways from this paper. But obviously I need to talk about other projects in this exit seminar as well. I do want to also shout out a companion paper written by Michael Hartigan, which examines the strawberry germplasm from a genomic standpoint, but has a lot of cool takeaways and results which dovetail nicely with this paper, which is from a pedigree perspective. And so with that, I'm going to switch gears and talk about the disease resistance work I've done while I've been here. As a quick primer, strawberry has had a really heavy reliance on chemical fumigation since the 1960s. If you look at the acreage in California planted since 1965, about 90% of that has been fumigated. Now, key ingredients of that fumigation mixture have faced international phase out over the years, specifically methyl bromide. But that and changing laws and regulations regarding what chemicals are allowed in what quantities and less effective chemical alternatives to methyl bromide have really affected the relative efficiency of fumigation as a primary control measure for soil-borne pathogens. And that's not to mention the growing acreage of organic production which can't use fumigation in the first place. And so because of this, you're starting to see novel pathogens emerge and historical pathogens reemerge as threats. One of the historical pathogens is verticillium wilt. This is caused by Verticillium dahlia, which affects many other crops as well, but it really is a major threat to strawberry. It's a century-old threat to strawberry and was actually one of the major drivers for the adoption of fumigation in California in the first place. Now, when we first started this project, we were looking at the literature and we noticed that most of it was restricted to prior to 1970. And what we realized is that as fumigation became more widespread, there was a deprioritization for breeding for verticillium wilt resistance and also for research into verticillium wilt resistance. And so we didn't have a lot of more modern literature to rely on. And the literature that we did have was somewhat outdated and somewhat ambiguous. And so we basically were starting from scratch in some ways. So in order to look at verticillium wilt resistance, we started with a global survey of strawberry germplasm. We looked at 984 different accessions planted in a randomized complete block design at the Armstrong pathology fields. And all of these plants were replicated 4 times and inoculated with a California isolate of verticillium. About 70% of the material we looked at was from the UC Davis program, but the rest consisted of heirlooms from around the world, as well as wild strawberry ecotypes from around the world. And all of these were scored on a 1 to 9 ordinal scale. And here you can see the distribution of scores over time. In the first year, we screened all of the material, and we downsized for the second year, with 388 total sessions being screened for both years. At the final time point for both years, we did see these bell-like curves, which is generally indicative of a quantitative trait. We also saw high broad sense heritability and a strong or a moderate correlation for the phenotypic resistance between years. So basically, what was resistant in one year remained resistant across years, and what was susceptible in one year remained susceptible across years. When we started pairing the phenotypic data with the passport data, we came across several interesting trends. One of these was that average resistance has appeared to decrease over time, whether that is in the wider diversity panel, so those 900 accessions, or just in UC Davis cultivars. And we think that there may be different explanations for this. If we look at material bred before 1950, before fumigation really began being looked at as an option to control verticillium wilt, we believe there may have been a bias for selecting resistant material then, because genetic resistance would have been the primary method of control. For material bred or past 1950 and 1960, we believe that fumigation might have acted as a crutch and allowed for a higher tolerance of susceptible material to be proliferated out into the industry. Another interesting trend we found was that there was a higher proportion of resistant material at more northerly latitudes. We believe that the climate at these more northerly latitudes is more conducive to verticillium wilt infection based on the biology of that pathogen. And so we think that might have led to higher rates of infection and host pathogen coevolution. When we looked for marker trait associations for resistance, we didn't find any. And then just to orient people who may not know what this type of plot is, this is on the x-axis, you see the strawberry chromosomes, and on the y-axis, you see a measure of significance. So the higher the value here, the more significant any one single DNA marker is in terms of its contribution to resistance. And so we didn't see any markers past the significance threshold. However, we were still optimistic because genome-informed breeding approaches such as genomic prediction have been used successfully for these type of traits in other economically important crops. For those who don't know what genomic prediction is, basically you're taking observations from the field, pairing that with genotypic data for the individuals you have observed in the field, and creating a prediction equation. When you look at the components of this equation, what you observe in the field is expensive and time-consuming to get, whereas the genotypic information can be cost-efficient and fast, and the creation of the prediction equation free if you have a computer, and really fast to create. And so the idea of genomic prediction is once you've collected enough data to create this prediction equation in the first place, you can start using the prediction equation in combination with genotypic data to predict what you would have seen in the field, predict what is expensive and time-consuming to look at in the field. So in order to assess the viability of genomic prediction for verticillium wilt resistance, we looked at 3 genomic prediction models. And for each model, we use 80% of the data to train the prediction equation, which then was used to predict the remaining 20% of the data. And we did 10,000 repetitions of this, randomizing the training and testing sets with each randomization. Here you can see the range of accuracies we achieved with our prediction equations. I do want to say that this range contains both the single year or within year, across years, single year use data, and combined year use data. So most of the accuracies here were towards the higher end of the range. And what this translates to is a relative efficiency compared to phenotypic selection of about 0.71 to 0.74. So that's really good when you consider that genomic selection can be used at the seedling stage. It can be used much more often than phenotypic selection can be used. These are kernel distributions, kind of very fancy scatter plots for our predicted scores versus our observations from the field. And what I really want to point out is that what we predicted to be the most resistant was the most resistant stuff that we observed in the field. And so we were really happy with these results. And so to tie up the verticillium wilt story, we show that genomic prediction has potential to accelerate the breeding of verticillium wilt resistant cultivars. And this is really important given how slow, difficult, and expensive that trait is to phenotype out in the field. But I also want to highlight that we've provided characterization data for over 900 different strawberry accessions. And so that's a resource the broader strawberry community can use to identify material they can use for future breeding efforts. And so this paper is already published. And so if you're interested in more details, feel free to go check out this paper. The other disease I want to talk about is actually the first one I looked at when I came here to Davis, and that is FUSARIUM wilt. It's caused by Fusarium oxysporum formispecialis virgeriae, and it was actually only 6 years old when I came in as a master's student because it was first described in California in 2009. It had appeared in other countries before, but when we looked at the international literature, there was a lot of argument whether the resistance people were seeing was qualitative or Mendelian or quantitative. So for my master's work, I looked at UC Davis material spanning the entire program's history. And these were planted at the Armstrong Pathology Field here in Davis and inoculated with a Californian isolate of the pathogen. We also looked at 2 mapping populations that had been derived from resistant cultivars. And you can see 2 of those progeny here. And so To make a long story short, because I do want to talk about the follow-up work I've done for my doctoral degree, we did see a high broad sense heritability, and this was due to the presence of a dominant Mendelian R gene, which we've termed FW1. And this R gene was confirmed in both the mapping populations via QTL mapping and across the broader panel with association mapping. At the time, we only had access to a SNP array that was based off of the diploid genome of the woodland strawberry. So we had localized it to chromosome 2. But one of the very first things I had to do for my doctorate was localize it to the octoploid genome, where it localized to chromosome 2b, which was previously known as 2,4. So launching into my doctoral work, my next major goal for FUSARIUM was to identify additional novel resistance genes. And a sub-goal of this was to develop markers for any novel genes we found to ease deployment via marker-assisted selection. So the reason we wanted to find novel resistance genes is that when you have a single resistance gene and deploy it en masse, you're putting pressure on the pathogen population, and that can lead to the emergence of novel races. So in this case, it would lead to the emergence of a race 2 capable of breaking FW1. And we didn't want that. So the whole point of this was to build up the breeding toolbox we have for Fusarium wilt resistance, have more options for resistance genes, and start using them in cultivars. To identify novel resistance genes, we looked at a broad diversity panel containing 344 different strawberry sessions. And so while my master's work had focused on UC Davis material, this panel was wholly composed of heirlooms and wild Frigeria ecotypes from around the world, capturing the broad diversity that exists. And these were assessed much in the same way as my master's work on RCBD out at Armstrong pathology fields and inoculated with a Californian isolate. We had really nice results in that we found that resistance was relatively common. 66% of the accessions we found were resistant to Fusarium wilt, and that was a large proportion of the cultivated strawberry as well as a pretty nice proportion of the wild strawberries. Here you can see the final phenotypic distribution, which is heavily skewed towards resistant material. And we also found that the resistance we were seeing was broadly heritable. You can see the broad sense heritability is 0.81, and it was very consistent across replications. However, one thing we didn't see was an association signal consistent with a single Mendelian R gene. And so what we believe is going on here is that while the phenotype we see in the field is common, Fusarium wilt resistance judged by eye, we believe that the genetics underlying that are actually caused by numerous rare alleles, and that any single one of these rare alleles is not present enough at a high enough frequency to be detected by this sort of approach. However, that didn't stop us. What we did is we pulled up a list of phenotypically resistant material, and we also looked at the genetics underlying the genetics of that material to see if they had FW1 or not. And so we chose 2 heirlooms and 2 wild strawberries to follow up on because they were resistant but did not appear to have FW1. For the heirlooms, we constructed self populations. These were Guardian and Wiltgard. And for the wild strawberries, we constructed outcross populations by crossing them to a common susceptible elite parent. And these were a Virginiana from Vermont and a Chiloensis from Chile. When we screened these populations, we were very happy to see segregation of resistance. In all cases, we saw bimodality. For Guardian and Wiltgard, we saw a 3-to-1 ratio. Considering that those were self populations, we found that to be consistent with a Mendelian resistance gene. segregating in those populations. And then for the populations derived from the wild strawberry, we found they segregated in a 1-to-1 ratio, which is also consistent with the segregation of a dominant resistance gene if the contributing resistance donor is heterozygous. And so this is just an example of the type of segregation we were seeing with plants that were really resistant and plants that were really susceptible. And this picture is actually from the population derived from the Fragaria chiloensis. And so when we did our genetic mapping and QTL mapping, we were pleased to see that our hypothesis was correct, that there were Mendelian R genes in control of the resistance in these populations. What was interesting is FW2, which came from the guardian heirloom, and FW5, which came from the chiloensis ecotype, were on chromosome 2b. Now, as a reminder, FW1 was also localized to chromosome 2b, and it turns out that FW1, FW2, and FW5 occur as an R gene cluster on the telomere of chromosome 2b. On the other hand, FW3, which is from the heirloom wilt guard, and FW4, which is from the Fragaria virginiana ecotype, exist on chromosomes 1a and 6b, respectively. For all of these resistance genes, they explain a moderate to high proportion of the variants, and they also were near dominant or completely dominant. The next logical step after these results was to design markers that we could use for marker-assisted selection. For this, we used a tool called PolyOligo, which was created by Mirko Letta, who was a postdoc in our lab. And so we're actually hoping to get some information on that tool published soon. But what we found is the designed markers were 95 to 97% accurate for FW1 through FW4, and FW5 saw markers with an accuracy of 90%. And just as a note about FW5, I think the decrease in accuracy there was because I couldn't directly design for the peak marker. It was in highly repetitive sequence. But overall, an accuracy of greater than 90% and 95% is really good. And so I think this shows that marker-assisted selection using these markers could really hasten the recovery of cultivars sparing a single one of these markers or multiple of these markers in conjunction. And so that was my second major Fusarium goal achieved. But I do want to remind you why I had that goal in the first place. It was to provide alternatives so we could provide a durable resistance and avoid the emergence of new races of the pathogen. Well, it turns out we were a little bit behind the curve because it turns out that race structure already exists in Fusarium wilt for strawberry. Specifically, there are already race 2 isolates capable of breaking FW1. This was work that was started by Peter Henry in the Gordon lab, kind of in parallel when we were doing our race 1 resistance work. And what he was doing is he was testing both domestic and international isolates against various strawberry accessions. For example, accession 24, which is a UC Davis cultivar carrying FW1. And what we found, because I ended up collaborating with him on this, was that FW1 does indeed protect against the race 1 isolates, but completely fails against race 2 isolates, leading to plant death. And so this was really concerning to us, and we decided we wanted to see what is the story on race 2 resistance in strawberry, which leads to the third and final goal of my Fusarium project. And this was to look at race 2 resistance in strawberry and see if we could identify any marker trait associations. To do this, we looked at the material we had already previously screened to race 1. And from that material, we selected a subset of 144 accessions to screen also against race 2. We did this screening at the Controlled Environment Facility at UC Davis because those the race 2 isolate that we tested was from Japan, and we couldn't let that get out into a field or greenhouse. So we had to do it in a growth chamber. So all the red points here are what we tested. Half of that material was from the UC Davis program, but the remainder had been hand selected to represent the diversity that exists across the Frigeria germplasm, including heirlooms and ecotypes. So when we phenotyped all this material, we found that in general, there was a decrease in resistance when comparing race 1 to race 2. 58% of the total panel was resistant to race 1, while only 28% was resistant to race 2. Within the subset of material from UC Davis, the decrease was much more dramatic, with 44% resistant to race 1 and only 5% resistant to race 2. But that was kind of expected, given that this race 2 isolate is known to defeat FW1, which is the resistance gene found in UC Davis material. We were very happy to find that there was still a rather large proportion of material that was resistant to race 2, and that the majority of that material was also resistant to race 1. And we did not detect any marker trait associations, unfortunately. And we do believe that this is, again, a case of that rare allele hypothesis, where while resistance, while there is a common phenotypic resistance, the underlying genetics are due to rare alleles where a single one is not present in a high enough frequency to be detected using this approach. And so overall, the FUSARIUM Wilt project provided a lot of cool results and a lot of actionable results too, things that could be directly implemented immediately. So since 2015, over 900 strawberry accessions have been characterized for FUSARIUM Wilt race 1 resistance. And in that same time, 5 resistance genes have been identified and are now exploitable via marker-assisted selection. In that same time, we've looked at over 100 strawberry accessions for race 2 resistance. And while we haven't been able to identify resistance genes to race 2, this preliminary characterization data does serve a good base for future projects to start at and to study, to identify and study any genetic loci that may occur for resistance, and then to start bringing that into more elite material. And so in summary, for all of my projects, I've explored and described the breeding history of the cultivated strawberry, and I've provided to the greater strawberry community genealogical and passport data for further analyses and studies. I've also provided resources to the community in the form of disease resistance characterization data for both FUSARIUM wilt and VERTICILLIUM wilt. And so I think that's really useful for people to follow up on if they want to. And finally, I did show that should say enabled, I apologize, that genetic and genomic-based breeding methods do have a place in strawberry breeding, such as marker-assisted selection for FUSARIUM wilt, or genomic prediction for more complex pathogens like VERTICILLIUM wilt. And with that, I would like to thank you guys for coming to see my talk. I would also like to thank everyone in the Knapp lab, Steve for being a great mentor, Glenn, Bruce, Randy, and Michael for providing plant material expertise in the lab or field, and just the basic tools needed to do this research. I'd like to shout out to Mitchell and Mirko, as they were key contributors to the genealogy project, and then Alan and Nico for being key contributors to the FUSARIUM Wilt projects. I'd also like to thank the Gordon Lab, specifically Dr. Gordon and Peter Henry. Without them, none of the disease resistance work would have been what it's like today. I don't think we would have even half the results we have. And I'd also like to thank Dr. Brummer for being really the first professor I knew here at Davis, and really being kind of a key mentor as well. Brian Policier out at Armstrong, Lisa Brown for keeping us sport and ag students sane, and finally my family. So thank you guys.
Thank you, Dominique. Nice job. The floor is open for questions, everyone.
Can I ask a question? I think.
Yeah, that'd be great, Paul.
So how easy is it to go to change ploidies? Because you have to end up with the octoploid, but if you have a broader Fragaria germplasm, can you easily introduce genes from, let's say, a tetraploid or a diploid, even a diploid?
That's a good question. So the progenitor species Virginiana and Chiloensis are both octoploids. So it's very easy to breed with them. However, so this is a somewhat complex answer. There have been recorded lower ploidy Fragaria in the genealogy. However, we're not quite sure how easily the allelic diversity from those lower ploidy Fragaria can be transferred into the octoploid in the long term. There is a lot of theory behind it, but no one's looked at it really in a scientific way. And a lot of those pedigrees that use those lower ploidy Fragaria end up as dead ends anyway, or they do focus kind of on that interploidy hybrid as the final result. They don't see it contribute further into Ananassa.
Yeah, it's interesting, Paul. I think what Dominique found, I can't remember the exact numbers, but it's a, you know, there's been a lot of reports of diploids and unusual ploidy sort of combinations. They're in the genealogy, but when you look at what Dominique found in the records, it's often difficult to see if there was actual confirmation of transmission, or, you know, if there was, it's really murky. I'm just only aware of a few really verified reports in that sort of secondary gene pool. Yeah, which there's a lot of material out there, right?
Mm-hmm.
Yeah.
So do you have like unreduced gametes in Fragaria?
No, so I'll talk about a specific example. What you see in some European breeding programs is a interspecific hybrid called the Fragaria X vescana. That's actually using Fragaria vesca to cross to Fragaria ananassa, Fragaria vesca being a diploid. Now what they do is they expose it to colchicine to make it a tetraploid, and then they do rely on unreduced gametes to end up with a decaploid after a few rounds of breeding. But they do focus on that decaploid as the final product. They don't really try to breed that back into Ananassa.
Yeah, so a lot of them are these sort of orphan pedigrees and dead ends, Paul, and you don't really see them ever get into that sort of main body. Also, just curiously, and I'm not an expert on the literature, I've looked at some of Bringhurst's old papers, but he found these natural pentaploid hybrids along the coast, kind of recorded those, and did a lot of really kind of cool early work on that. It was very interesting. Because the diploid and octoploid species are sympatric in some of these coastal zones, and they form natural hybrids. Yeah,
if you're interested in reading more about that, there's a lot of theory out there revolving around, I think they call it synthetic octoploids in strawberry, and they basically lay out these theoretical roadmaps on how you would make an octoploid out of a diploid or a tetraploid and try to bring that into strawberry, the cultivated strawberry. But as far as I know, no one's really followed up on that theoretical work.
I had a question if you're
Yes, please.
okay. Before I arrived at Davis, I was also doing some archival work trying to track down the work of a breeder in a different, completely different area. And at least in my case, you know, I ended up having to go to lots of different archives because his notebooks didn't end up at the same place. And I was wondering if because, you know, Davis has such a long-standing strawberry breeding program, if it was the sort of thing where you just go to special collections here and you were, you know, it was all waiting for you, or did you really have to dig around?
No, so basically, if we look at the physical records that walked away from the university, there was two major ones. The breeding notebooks of Thomas and Goldsmith, who were the first breeders here at Davis, and Bringhurst and Voth, who were the breeders at Davis from 1950 to 1990. And so in both cases, the records had stayed together. They hadn't spread out. One had ended up in a private collection, and the other one had ended up in Utah, actually, donated by the son of Bringhurst to Utah State, Logan. So I did have to do a lot of traveling. I basically lived in Utah State's library for a week, transcribing all of these. But yeah, we were very lucky in that those records had stayed cohesive and together.
And that the private collector, I guess, was willing to let you look at what they had. They weren't just, you know, being.
Yeah.
Well, the notebooks were at Driscoll's.
Oh, okay.
Oh, I wasn't sure if I was allowed to say that or not.
You are. We acknowledge that. And I, you know, the breeder said, yeah, I should have given these to you, but Dominque actually had to go there and, you know, basically photograph the books so we could get those records. But we're fine. It was one of those weird discoveries, you know.
Yeah. Well, it's great that it was able to come to pass that way, because sometimes some things get into private hands and stay in somebody's home, you know, until you reach a relative who doesn't care about them anymore.
Right.
And I also, you know, I missed the first couple minutes because I had difficulty logging in, so I may have missed this information. But if somebody wants to read your genealogy of strawberry history, where would they go to do that, or, you know?
Oh, so that was just accepted by G3. So we expect it to be, we expect an advanced publication to be available within a month, and the official publication to be within a month or two.
Okay.
But we can also send that to you directly if you send me an email.
Just send her an email. She'll get it to you.
Well, thanks very much. It was a great presentation, really interesting.
Thank you.
Any other questions for Dominque? Okay. Well, Dominque, congratulations, and really, really proud of what you've accomplished and appreciate your seminar today. And I thank everybody for coming in and joining to celebrate with Dominque today. Wish we could do it in person, but I don't think that's possible for a while yet, Dominque.
No, I totally understand that. But thanks for coming out and checking out my projects today, everyone. Yeah. And if you have any questions or want to follow up on some of these, look at the publications, or feel free to reach out. I can direct you as needed.
Great. Everybody have a wonderful holiday, safe and wonderful holiday, okay?
Thank you, everyone.